everyone, it's me Shanty and you are watching Biology Nowadays. In this video, we will learn about phloem, which is one of the complex permanent tissues present in plants. As you already know, xylem and phloem are the two complex tissues found in plants. Xylem tissue is specialized for water transport, whereas phloem tissue is specialized for food transport. These tissues act as a continuous channel for the conduction of water, mineral elements and prepared food. In the previous lecture, we already discussed about xylem, its components and functions. In this video, we will learn about phloem. The name phloem is derived from the Greek word phloios, meaning bark. In trees, phloem is the innermost layer of the bark. Phloem is also called as bast. Like xylem, phloem also consists of tube-like cells for food transport. In phloem, food is transported in the form of a water-based solution called the phloem sap. Food is basically the carbohydrates prepared by plants through photosynthesis. You know that plants prepare glucose during photosynthesis. In most plants, this glucose is converted into sucrose for transportation. You may be wondering why plants use sucrose for transportation and why not glucose? The first reason is that sucrose is more efficient in energy storage when compared to glucose. And the second reason is that sucrose is not as much reactive as glucose. Because of these two reasons, most plants use sucrose for transportation. So we can say that phloem tissue transports food in the form of sucrose, usually from leaves where the food is prepared, to other parts of the plant for storage and growth. This means that the flow of food in the phloem is downwards. But you know, in winter, the temperate plants shed their leaves and in spring, new leaf buds start to appear. These leaf buds require food to grow into fully mature leaves. During this time, the food materials stored in the storage organs such as roots are used up. Then, the food flow in phloem is from roots to leaves, that is, upwards. In short, the movement of food in phloem can be bidirectional, downwards or upwards. But in the case of xylem, the water flow is always unidirectional, that is, from roots to other plant parts. As in the case of xylem, phloem is also composed of 4 different kinds of cells: sieve elements, companion cells, phloem parenchyma, and phloem fibres. Sieve elements consist of sieve tube members and sieve cells. But unlike xylem, phloem is a living conducting tissue. Except phloem fibers, the other 3 components are living. Do you know why xylem cells are dead and phloem cells living? This is because the processes that help in water transport in xylem are passive, which means that they do not require an input of energy, that is ATP produced inside a living cell. On the other hand, the phloem transport is at least partially an active process. That is, it requires the input of energy in the form of ATP. So, the food conducting phloem cells have to be living. Among the phloem components, sieve elements are the actual food conducting cells. First, let's learn about sieve tube members. Sieve tube members are the advanced food conducting cells and they are seen in the advanced plant group angiosperms. Sieve tube members are short, cylindrical, tube-like structures. They are arranged vertically, that is, one above the other, to form long tubes called sieve tubes, just like the xylem vessels. The end walls of the sieve tube members have pores like a sieve. So now you know how they got their name. The end walls of the sieve tube members with sieve-like pores are known as sieve plates. There are two types of sieve plates, simple and compound sieve plates. In a simple sieve plate, there is only one sieve area. Sieve area means a cluster of pores. In a compound type, the sieve plates have several sieve areas. Actually, the sieve tube members have some sieve areas on their side walls also. But the pores in these sieve areas are really small when compared to the pores on their sieve plates. The sieve tube members with single sieve plates are more advanced. The pores in the sieve plates are actually modified plasmodesmata. Plasmodesmata are small channels that directly connect the cytoplasm of neighboring cells to each other. In the sieve tube members, the living contents of the cell or the protoplasm is distributed as a thin layer along the walls so that there is no block to the flow of food materials through these cells. Now be ready for a surprising fact. Even though sieve tube members are living at maturity, they don't have a nucleus. That's interesting, right? Without a nucleus, who controls the activities of these cells? Sometimes we say that if we have a good companion, many problems in life can be solved. In the same way, each sieve tube member has a good companion throughout its life, called a companion cell. The companion cell has a nucleus and it controls the metabolic activities of the sieve tube member. Each sieve tube member and the associated companion cell develop from the same mother cell. The relationship between them is so close that when the sieve tube member dies, its companion cell also dies. Before we get into more details about the companion cells, let's discuss about the second type of food conducting cells, the sieve cells. They are elongated and narrower than the sieve tube members. They are primitive food conducting cells and are seen in primitive plant groups such as pteridophytes and gymnosperms. Instead of sieve tube members and companion cells, these plant groups have sieve cells and albuminous cells. The main difference between a sieve tube member and a sieve cell is that the sieve cells have sieve areas throughout the side walls and also in the end walls. But as I mentioned earlier, the sieve tube members have sieve areas mainly in the sieve plates. Sieve cells have smaller sieve pores than that of the sieve tube members. Each sieve cell is connected to an albuminous cell. Albuminous cells are just like the companion cells in all aspects, but there is one difference. The sieve cell and its albuminous cell don't develop from the same mother cell. Now let's see more about the companion cells. The companion cells are specialized living parenchymatous cells which are closely associated with sieve tube members. They are connected to the sieve tube members by plasmodesmata. In a longitudinal section, you can see that they are elongated, narrow cells. Now let's see how they look in a cross section. In this cross section of a monocot stem, you can see that xylem and phloem are seen as an organized group. This is the phloem region and these are the xylem vessels. The xylem and phloem is protected by a layer of sclerenchyma cells. Let's focus on the phloem region. The bigger cells that you see here are the phloem sieve tubes, and the smaller cells are the companion cells. Sometimes in a cross section you can also see the sieve plates of the sieve tubes. The companion cells have dense cytoplasm with a prominent nucleus. They have large numbers of ribosomes and mitochondria. ATP produced in these mitochondria help in providing energy for translocation of food materials in the sieve tube members. Now let's move on to phloem parenchyma. They are the parenchyma cells associated with phloem. They are living, thin-walled, elongated cylindrical cells with tapering ends. They have dense cytoplasm and a nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food materials and other substances like resins, latex and mucilage. Phloem parenchyma is present in pteridophytes, gymnosperms and dicots. It is absent in monocots. We already saw how phloem is arranged in a monocot stem. Phloem in monocot stems consists of only sieve tubes and companion cells. Phloem parenchyma cells are absent here. Shall we have a look at the phloem in dicot stem? Let's analyze a part of this tree stem which shows secondary growth or thickening. The anatomy will look like this. I took this section during my wood anatomical studies. This part is the secondary xylem or wood, and here is a xylem ray. This narrow region contains the vascular cambium, which produces secondary xylem and secondary phloem. And this is the phloem region. Secondary phloem lies here. And these groups of cells are the primary phloem cells. Like the primary xylem, primary phloem cells also arise from the procambium of the apical meristem. The primary phloem cells are usually crushed and functionless in trees due to the formation of secondary xylem and secondary phloem tissues. Like in the case of secondary xylem, secondary phloem has two types of parenchyma, namely phloem parenchyma and phloem ray parenchyma. Phloem parenchyma cells are elongated in the vertical direction and ray parenchyma cells are elongated in the radial direction. From this cross section, it is difficult for us to correctly spot the phloem parenchyma cells because the sieve tubes and the companion cells are also thin-walled like parenchyma cells. But you can easily identify the phloem ray parenchyma cells as they are radially elongated cells. Besides storage of food materials, phloem ray parenchyma also helps in lateral conduction of prepared food materials. Now, what are phloem fibres? Phloem fibres are the sclerenchyma fibers associated with phloem. They are also called bast fibres. They are found mainly in the secondary phloem. Let's come back to this picture. Here you can see a long band of phloem fibers in the secondary phloem region. But in the primary phloem, they are present in fewer numbers or sometimes absent. They are much elongated and unbranched. They have tapering ends. The cell wall of phloem fibers is very thick and so they have narrow lumen. At maturity, these fibers lose their protoplasm and become dead. They provide mechanical support to the plant as well as protect the phloem tissue because phloem cells are thin-walled. Phloem fibers are extracted from the bark of plants like jute, flax and hemp for making threads, ropes and textiles. Now I hope you got a clear picture of phloem, its components and functions. Finally, let's see what is protophloem and metaphloem. The first formed primary phloem cells are called protophloem, whereas the later formed primary phloem cells are called metaphloem. Metaphloem has bigger sieve tubes, whereas protophloem has narrow sieve tubes. That's all about phloem. But before I conclude this lecture, I have an interesting question for you. Suppose that I removed a ring of bark from the base of a tree, just like this. This process is called girdling. Now the question is, how will it affect the food transport? Will the tree be able to survive? Type in your answers as comments, and that's all for this video. Thank you very much and stay curious.